All right, why don't we get started? So it's great to have the last OneNeuro speaker series for the year. We'll start up again in the fall. And I wanted to call your attention to our recently launched website shown here. And I don't know if any of you have seen it, but just do OneNeuro, no space, Hopkins, and you'll find it. And so it has a lot of content. It has a lot of content, including links to, recent papers coming out. There's interviews with 2 faculty so far, so a lot of information. Calendars. We're trying to sync all the calendars in neuroscience department related departments. We're going to have announcing the Discovery Awards that are the OneNeuro supports in collaboration with the provost office, hopefully, who knows, in the next few weeks or so. And so the links to education, schools, et cetera, et cetera. So take a look. And I would go there often. Every other week, we're going to present an interview with a faculty member. And we have some colorful interviews, talking both about science and personal hobbies and things like that. So I will turn it over to Tricia, who's going to introduce Fred. And if we can get out of here.
Thanks, Rick. Hi, everybody. Thanks for coming. It's also so nice to see lab members. So this is not just for community building for faculty, but for all the different people in our different labs to get together, and undergrads as well. So it's my pleasure to introduce our first speaker, who's Dr. Fred Barrett. So Fred is a professor in the neuropsychopharmacology of consciousness. That's about the coolest job one could ever have. He's a professor in the Department of Psychiatry and Behavioral Sciences at the School of Medicine, and also has an appointment in Psychological and Brain Sciences here at the Krieger School. And he's the director of the Center for Psychedelic and Consciousness Research. And now, Dr. Barrett's a cognitive neuroscientist, but has strong training in behavioral pharmacology, very relevant to our topic today, and has been conducting research on psychedelics since 2013 here at Hopkins, both in healthy humans and also in humans who suffer from mood disorder or substance use disorder. And so now Fred's going to tell us about psychedelics in humans.
Yeah, thank you, Rick, and thank you, Trish, for the wonderful introduction. I'm going to talk to you today about psychedelic drugs. Primarily, we study psilocybin, which is the active component in literally hundreds of species of mushrooms that grow around the world. I'm hopefully going to get into a high overview of some data, but I want to start at the beginning of asking, well, first, letting you know that I've collaborated and advised with some industry partners. Nothing that I've done in that space will be what I'm talking about today, but I should disclose these things. But then again, starting at the beginning with what are psychedelics? If you look in the dictionary, you'll find a definition that includes words and phrases such as hallucination, sensory perception, perceptual distortions, big feelings. But these are, I would argue, of maybe the least or the less interesting effects of these drugs. The word psychedelic was coined by a British psychiatrist who was expatriated to Canada and started one of the first clinics that administered LSD in the modern era, or I guess ever, in a clinical setting. Humphrey Osmond administered LSD to patients in his alcohol use disorder clinic. He also consumed LSD on his own, as well as psilocybin and mescaline, which is found in many psychoactive cacti. Osmond took these drugs to try to figure out how they worked for him, and maybe then he would have some insight in how they may be useful in the clinic. And he believed that these compounds somehow revealed something about the mind that was otherwise impenetrable or not accessible during normal waking consciousness. And so from that, he coined this phrase, psyche meaning mind and delos meaning manifest, mind manifesting. And if you take that at the most generous interpretation, the coffee I had this morning was mind manifesting, but I don't think that's what he was really referring to. There's something kind of bigger and more prominent and pronounced than that, which we'll get into a little bit. But just to kind of give you a tour too, psychedelics are thought to have been used in a number of indigenous contexts for many thousands of years, if not longer. There are mushroom statues that have been found throughout Mexico and evocative cave paintings suggestive of a shaman who's covered in mushrooms, possibly using these for ceremonial purposes. More recently, and carrying into the present, there are some native North Americans who have used peyote cacti in ceremony for quite some time. In the Amazon, you have ayahuasca, which is a plant admixture formed of a vine that has Banisteriopsis caapi, which has dimethyltryptamine within it, and another plant that essentially Psychotria viridis is one of them. There are actually a number of different combinations that people can come up with to put this brew together, but essentially dimethyltryptamine is a tryptamine psychedelic that gets broken down by monoamine oxidase in the gut. And so at least many hundreds of years ago, if not further, natural psychopharmacologists in the region figured out if you mix this thing with the other thing that happens to have MAOIs in it, monoamine oxidase inhibitors, and you ingest that, the MAOIs inhibit the MAO in the gut, and so you can ingest it, it doesn't get broken down, it can become systemic and pass into the brain. And it's remarkable that people figure these things out. So things from way past peyote, ayahuasca, mushrooms, and then we have many laboratory chemicals that were synthesized first in the laboratory like LSD and MDMA. So that gives you an idea of just, broad strokes, what are psychedelic drugs? The way we use them now in the laboratory is very kind of particular. These are never going to be take two and call me in the morning type drugs, at least if they're ever approved as medicines. But when we use psychedelics in the lab, and this may inform how they're used in the clinic, if they're ever approved as medicines, we go through careful process of screening out people with cardiovascular risk, not because we know that psychedelics necessarily have a specific cardiotoxic effect, but because they are generally known to increase heart rate and blood pressure in a stereotyped way. And if you increase the person's heart rate and blood pressure and they have some cardiac vulnerability, you can stress that and cause problems, so we don't want to do that. Psilocybin, there are many drugs that fall under this umbrella of psychedelics. Psilocybin itself from mushrooms is actually rather physiologically safe. And I say that with some caution because nothing is totally safe. Nothing's ever totally safe. But they've tested for the LD50, the lethal dose of psilocybin in many animal models, and the scaling and the translation there would suggest that you'd have to take many thousands of times the therapeutic dose of psilocybin for a human in order to reach one of those kind of possibly physiologically lethal doses. That's not to say that these are not without risks. They absolutely with risks, but if you think about the therapeutic window, that's the difference in the dose that helps versus the dose that hurts. The therapeutic window of aspirin is quite small. You can take enough aspirin to really hurt yourself without too much effort. You'd have to really work hard to take a dangerous dose of many psychedelics, but that's physiologically. The real risks of psychedelics come from a psychiatric and psychological perspective. Briefly, we very carefully screen out people who are at personal or family risk of psychosis or bipolar disorder, and there are a number of other indications that may present a particular psychological risk to participants. But once we've screened these folks very carefully, we then undergo a long period of preparation whereby we try to build trust and rapport with the participants. We try to help them understand that they're in a safe place and they can be free to experience whatever they need to. We also try to counsel them on what may occur during psychedelic experiences. And this is very important because we can never predict exactly what will occur to any individual on any given day, even with a known dose of a psychedelic drug. After all of that occurs, we then invite a participant in for a drug administration session. We invite them early in the morning. We do a final screening and then administer a capsule, and then we monitor participants for 4 to 6 hours after that capsule administration. With psilocybin, that's about the time course of drug effect. By the end of the day, they're back, they're healthy, they're on their way home in the arms of a loved one, and then they come back to the lab for days and weeks and months afterwards for continuing aftercare and integration, continued therapy to help people make sense of these experiences. I wanted everybody to understand that because when we talk about psychedelic-assisted therapy, that's what we talk about, that whole package, that container, that process that involves a lot of careful preparation, monitoring, and then a lot of aftercare and integration. So what? Why are we so excited? Well, there have now been a number of clinical trials published, some small, some getting a little larger, in which we're seeing remarkable therapeutic effects of psychedelics. Those therapeutic effects, this is the first ever randomized clinical trial in patients with major depressive disorder, and demonstrating a remarkable therapeutic effect in admittedly a rather small sample. 24 individuals were administered 2 doses of psilocybin. And then from this initial time point here, the pre-dose baseline, this is a clinician-rated, a blinded clinician-rated depression severity scale. Above about 8 is mild to moderate, about mild, moderate, and severe. So patients in this range were moderately to severely depressed upon entry. And then after 2 doses of psilocybin, had a precipitous drop into the range of remission. Over half of the participants in the study were in remission from depression for at least a year after 2 doses of psilocybin. This is the pretty average that we like to put as the first figure, and this is the dirty reality, a spaghetti plot. Each line represents one subject, and you can, if you squint your eyes a little bit, you can see, well, yeah, actually, most folks had a precipitous drop in the depression severity and stayed low for the most part, but there are some conspicuous outliers. This person at the top was clearly a non-responder and the most severely depressed when they entered the study. And then there are a few folks in the middle who got better and then kind of bounced back up to baseline levels of severity. So a few takeaways. We shouldn't expect this to work for everyone. There is individual variability in response, and there are some more things to unpack. This is not a direct follow-up to the study I just showed you, but we were one of the sites in a multi-site clinical trial of nearly 100 people who were randomized to receive either psilocybin in that container that I told you about, or a very high dose of niacin as an active control of sorts. And we see, hardly any reduction in depression severity after niacin administration, but a remarkable reduction after psilocybin. I think over 42% of people in this study were in remission from depression for at least 43 days after, about 6 weeks after administration. One of our colleagues at Imperial College London did a head-to-head comparison of a placebo psilocybin session, so the whole package, but a sugar pill, and then daily escitalopram, a standard first-line SSRI treatment for depression. Placebo, active escitalopram for 6 weeks, or active psilocybin, and then daily placebo pills for 6 weeks. And the primary outcome here was depression severity compared between conditions at 6 weeks. And that was the pre-registered outcome. And some tried to say, clearly this was a failure of a trial. You didn't see a difference in your primary outcome. But I think the more reasonable interpretation is at least a non-inferiority outcome. We're talking about 6 weeks of daily pharmacotherapy compared to 2 days of pharmacotherapy, and an equal outcome. And these are this was the pre-registered primary outcome, but a number of secondary outcomes are either equal or favor psilocybin, some strongly in measures of depression, anxiety, well-being, things like this. When looking at treatment-resistant depression, we see a similar effect. A single dose of psilocybin showing a remarkable reduction in depression severity in patients who were treatment-resistant. And in this study, 37% of people were in remission after, 12 weeks, for at least 12 weeks. And then another study was published very recently demonstrating that between 1 to 2 to 3 doses, you see an incremental increase in therapeutic response. So this is why people are so excited. But this is not the only reason that people are excited. As I said, one of the first studies and one of the first uses in the clinic was for substance use disorder, specifically with alcohol. Most of the studies in the first era of psychedelic science between basically the 50s and 60s, there were hundreds, if not thousands, of studies and papers published. The vast majority do not meet any semblance of rigor and reproducibility that we would accept in current clinical trials. But these folks, Krebs and Johansson, found 6 out of the many hundreds that they felt they could in any way justify or were controlled. And these are 6 studies with various control conditions. One was treatment as usual. One was LSD compared to just a sugar pill. One was, take LSD or go into AA. But so with this grab bag of control conditions, a meta-analysis on these clinical outcome data favored LSD over the grab bag of control conditions from the previous era. A more recent study, both a 10-person pilot study and a much larger study of about 80 to 90 people, demonstrated either with an open label session or comparing psilocybin administration in the package to diphenhydramine, or basically Benadryl, in that same setting, a favorable response of psilocybin in reducing drinking in patients with alcohol use disorder. So these are co-occurring disorders. Oftentimes, people who are depressed are more likely to drink. People who drink too much are more likely to be depressed. It's the rule rather than the exception in real humans who are suffering in the world to have these co-occurring disorders, despite the fact that they're often studied in isolation. But with these parallel lines of evidence developing of potential for psilocybin, this virtually the same intervention to treat patients with depression or patients with alcohol use disorder, I am asking the question of, well, what if you combine them? What if you look at the combined co-occurring disorders? So I'm in the middle of running a study. We're almost halfway out of 90 participants enrolled. Patients have to be within a certain age range, have both major depressive disorder, a current depressive episode, a baseline of at least moderate depression severity, meeting criteria for alcohol use disorder, but not the most severely acute drinkers, not people who are at risk for complicated or medically complicated withdrawal. So people who are drinking too much, but not too much. Part of the reason being that these things can really reinforce themselves. People who are drinking and depressed are less likely to seek treatment. When they seek treatment, they're less likely to adhere to treatment and less likely to respond to treatment, less likely and more likely to remit after even a partially successful treatment. So we're trying to not make it too hard on ourselves here, but essentially this is a slightly complicated study design. At enrollment, we have baseline blinded depression severity and alcohol assessments. Then people go through this preparation period that I indicated. They have a short, brief motivational interviewing intervention for drinking, and then they enter the experimental phase where they're randomized to either placebo or a high dose of psilocybin. And then 3 months of follow-up over which we track depression, drinking, liver function tests, and a number of other things. And so after this 3-month period, this initial period, if they get past this point for all their primary outcomes, we're considering them completed, but then they have the option of crossing over to an open label psilocybin administration session, which is just mirroring that session and then long-term follow-ups. And so we're not going to talk about the experimental phase. It's unblinded, it's ongoing. I'm not going to touch it, but we know if people get to the open label phase, they've received psilocybin, so I feel a little more comfortable peeking at these data. And what we're finding is from that baseline right after enrollment to 1 month after the open label psilocybin session, we're seeing a remarkable reduction in depression severity. Lots of caveats here. So first, I mean, the Cohen's d is nearly 4, which is itself a little absurd. But I guess you don't really need a Cohen's d to see the experimental effect here. We don't know who received psilocybin twice versus once. We don't know what the trajectory of depression severity is in the middle here. There are lots of unanswered questions, which I really hope to be looking at in about a year from now. But also, remarkably, there's at least one non-responder across this entire intervention, and some folks who respond a little less strongly or a little more strongly. This is depression severity. The same patients are showing a reduction in heavy drinking days over the past 90 days and days abstinent over the past 90 days. And also, we're collecting a number of different objective biological measures that can be associated with severe alcohol use. And this is one liver function test that is sensitive to about weeks to months worth of drinking. And some folks have low liver function tests to begin with, low evidence, so low to no evidence of liver damage. And these are folks who are on the lighter end of drinking in our sample. And not surprisingly, those who are drinking more have more severe kind of measures on this liver function test. And there's a nonlinear relationship here. If it ain't broke, you're not going to fix it in a sense, right? But positive sign here, we still have more data to collect. There are a lot of questions embedded in here. There's some folks who believe that due to heavy alcohol use, patients may need higher doses of the drug. There's evidence that folks who are drinking too much may need more anesthesia if they're going to surgery, more antibiotics if they need treatment for some infection and things like this. We're actually seeing, usually when you give someone either a very high dose of psilocybin, 25 milligrams, which is roughly equivalent to 4 to 5 grams of dried mushrooms, which is a whopping massive dose of psilocybin, there's usually no question, either you have it or you don't. About 2 hours into the session, people know, and usually the study team knows, and it's a huge problem in the field because it's essentially functionally unblinding the experimental conditions, and the field's really grappling with it. There are a lot of folks who they're not sure, and we're not sure. It may be that there is an effect of alcohol blunting the effect of, not acutely, they're not acutely intoxicated, but having used alcohol for a long period of time is blunting the acute effects, but yet still people seem to be having some sort of therapeutic response. Another question is whether or not chronic treatment with selective serotonin reuptake inhibitors, SSRIs, SNRIs, or similar drugs These are thought to possibly change the expression of serotonin 2A receptors in some important parts of the brain, especially prefrontal cortex, where psilocybin and other psychedelic drugs are known to bind and have some of their effects. So there was a possible risk that SSRI use concurrently is kind of possibly going to blunt or reduce the effects or the efficacy. And now about half of our participants are maintained on SSRIs throughout this, and we're not necessarily seeing a differential effect. Remarkably, there was one patient who experienced no subjective effects during the open label session. There are a lot of questions that we need to dig in here, but again, we're not there yet. A lot of questions to ask about I want to make sure I'm checking my time. Oh gosh, okay. I'm gonna skip ahead. I took a little more time than I thought. Okay, so I'll take a little longer then. Okay, you tell me when. We have a lot of thoughts about how this all might be working. We're building evidence that psychedelics may possibly increase psychological, cognitive, and neural flexibility. There may be effects on episodic memory that are at play that are mediating therapeutic outcomes. There may be anti-inflammatory or neuroplastic effects, and of course, Goldolan, who's here, has done a lot of work in this. To dig into a lot of these questions, we're essentially taking people on magnet, doing cognitive and emotional memory tasks, getting spectroscopy and structural imaging before and after. We're taking people off magnet and doing another battery of cognitive tests, getting polysomnography, electrophysiological measures. We're studying the gut microbiome, and our study team has kind of lovingly called this the kitchen sink protocol. So we're essentially getting the kitchen sink on people right before and a week after a drug administration, so we can begin to see whether or not we're moving the needle on a number of precise things. And I want to dig a little bit into this. So we did an online survey of nearly 1,000 individuals who we said, if you've taken psilocybin mushrooms and then happen to spontaneously recover from depression, take our survey. And we had roughly almost 1,000 people complete this survey and provide us what we considered good data. We asked them all sorts of questions about their experience, how they felt before the experience, how they felt after it, what the changes were that they could remember. And of course, caveats abound. These are entirely retrospective data. We're almost certainly succumbing to some sort of response bias of people who had a good experience. We're not getting a prospective understanding of this. People who think they got better or recovered from depression after having mushrooms, they may have a positivity bias towards other aspects of the experience and may not remember fully, but from the data we collected, we were able to conduct a path analysis and demonstrate that, there are certain kind of really profound and peculiar aspects of the subjective experience during psychedelic experiences predicted, or at least were associated with the acquisition of psychological insights that were idiosyncratic. We would have never been able to predict them, but seem to occur at a high rate in people who have a therapeutic response. And this may be one of the active ingredients. It's having these insights and then carrying them forward. So people who encountered these insights were more likely to rate an increase in psychological flexibility after the experience that then predicted decreases in depression severity. So it wasn't anything about the acute effects of the drug or these acute insights directly that clearly and most powerfully explained the decrease in depression that people reported. We followed up in that study of 24 individuals, and we had them complete a cognitive flexibility task. This is a version of the Wisconsin card sort task. Essentially, you have these 4 figures on screen. You're told to click the object that doesn't belong, and well, which is it? Is it this one because it's small, or this one because of line weight, or this because of shape identity? And so you click an object, you choose. It tells you if you're right or wrong, and you click through and you try to find the rule, and then you find the rule. Oh, geez, it's line weight. Great, line weight, line weight, line weight, line weight. I'm good, doing so good. I'm getting it correct. Then all of a sudden, the rule changes without you being told, and then incorrect, incorrect, incorrect. Then you have to find the new rule, keep responding to the new rule. After a certain point, it changes again, and then you have to find the new rule, and the degree to which you continue to perseverate on the old rule before searching and finding the new rule, those are perseverative errors. Those are thought of as measures of cognitive inflexibility. And so to the degree that perseverative errors reduce after a treatment, you could interpret that as an increase in flexibility, and that's exactly what we found. So that study was a waitlist control design. Patients who were randomized to the delay condition did this task at baseline, waited 8 weeks, did the task again, and then engaged in treatment. You can see there are fewer lines here and more lines here because those who were randomized to immediate treatment, they just did the task, got treatment, did the task again. And what we see is overall, no clear evidence for a learning effect in this task, which itself is remarkable because it's incredibly susceptible to learning effects. But we didn't see it in this depressed population, which is also consistent with a deficit in cognitive flexibility in patients who are drinking and depressed. But what we're seeing here is an increase in cognitive flexibility from before to after treatment, and also an increase in the dynamic connectivity of between the anterior cingulate and posterior cingulate, which anterior cingulate being implicated briefly and broadly in various aspects of cognitive control, posterior cingulate being variously in broad strokes involved in certain aspects of self-reflection, self-identity, and self-referential processing, and the posterior cingulate has been implicated in the acute effects and the enduring effects of psychedelic drugs. So if I can completely overinterpret this, we're seeing evidence for changes in psychological flexibility, cognitive flexibility, and possibly neural flexibility, if you'll allow me that, which they're all actually correlated to some degree with antidepressant effects. And finally, to close it out, I think there may be a story for the claustrum, not colostrum. Sometimes people get confused because I mumble, but I've been working with folks at the University of Maryland, specifically Brian Mathur and his lab, who have been deeply studying the claustrum. It's an area that densely expresses serotonin 2A receptors, which are the apparent target of psychedelic drugs. The claustrum has reciprocal connections to nearly all areas of cortex. And actually, Human Kadir did a remarkable task of studying over 1,000 corticoclaustral circuits with, I think, keep forgetting, ex vivo circuit mapping, thank you, during his grad student tenure. And he found a number of kind of circuits and motifs, as well as two types of claustrum neurons, a burst firing and a single firing claustrum neuron. And essentially what he found was that, given a certain network state space, this is the story that, given a certain network state space, if you need to switch network state space in order to respond to changing demands in the environment, what happens is the claustrum, the type 1 claustral neurons will fire, kind of send out a signal that breaks up that cortical network state space, and then claustrum type 2 neurons with burst firing will come on in order to kind of synchronize and bring online that secondary network state space. And the story here briefly is that psychedelics may disrupt the function of the claustrum in managing and regulating network state space in the brain. We have some first-in-human imaging, resting state imaging findings to demonstrate or suggest at least that psilocybin may disrupt frontoparietal task control network as well as default mode network function, which may lead to, here's the punchline, and I'm going to give you the briefest explanation of this and ask you to read my and Dr. Jada Sayala's recent review on this in Neuron, but imagine depression and substance use disorders as illnesses in some form of a lack of cognitive flexibility in rumination, being stuck in rumination, or being stuck in repetitive drug-seeking behaviors. And imagine these in an unbalanced network or cognitive control strategy. We can think of meta control in cognition as the active and ongoing balance between flexibility and stability in cognition. And we can imagine plenty of instances in life where it's good to maintain some cognitive stability, like when I'm trying to write a paper, or when it's good to maintain some flexibility, like when I'm trying to drive downtown in Baltimore. And our ability to flexibly manage this is meta control. And if we think of depression and rumination in some part as an illness of lack of cognitive control, not cognitive flexibility, not being able to be flexible enough to think out of your negative, ruminative, self-referential thoughts, that we can think of substance use disorder in part as an illness of a lack of cognitive flexibility and not being able to break out of the identity of being a drinker or a smoker, or break out of this repetitive kind of drug-seeking behavior. This is an unbalanced control strategy to the degree that psychedelics radically unbalance in the other direction to an enormous amount of flexibility over flexibility, which may be expressed in some ways during the acute effects of the drug. It may be possible that also with the possibility, and now I'm really over speculating, but if there's some possibility for a critical period of learning to open up that would allow for flexibility in learning new kind of levels of control, it's possible that in this state, the brain may learn the value of flexibility or relearn the value of flexibility and end up in a long-term balanced kind of control strategy. And I kind of murdered that a little bit. You need to talk to Jada. She has a much more eloquent way of explaining this, and I'm also trying to rush through it, so please forgive me for any damage that I've done to these concepts. But essentially, you know, we are taking some of the kitchen sink data in a number of different paradigms on and off magnet to look at various aspects of cognitive control and cognitive flexibility and whether these shift, whether it's due to response inhibition, working memory, or other things. And this is just a list of most of the other studies that are being conducted at the Center for Psychedelic and Consciousness Research. We're really trying to explore and expand and find the edges of the indication space of what illnesses, psychiatric mostly, that these compounds might be helpful in treating, and then understanding as best as we can some of the mechanisms. I just want to plug some of the other work that we're doing. My grad student, Brian Winston, has a poster today where he's digging into some of these mechanisms and in collaboration with my colleague, Janice Chen in psychology. We're looking at video stuff. Don't stare too hard. Go to Brian's poster. And this is another poster that'll be led by my postdoctoral fellow, David Rosen, where we're trying to expand upon this idea of flexibility and critical periods in human models, really searching deeply for, are there any human models where we could really look and interrogate some of the incredible work that Gul has done in this. Don't stare at the poster, go talk to Dave. This is even yet a translational study in obsessive compulsive disorder that our postdoctoral fellow, Prachi Tiwari, is going to be presenting on with animal models and a human study that is nearly complete. Don't stare at the poster, go talk to Prachi. And there are two other posters, one from Emma Shea O'Donnell, Dr. Emma Shea O'Donnell, she's going to be, and the final poster is Brianna. So please see the posters afterwards. That's my plug. And I just want to thank all the wonderful people who work in the center. And I hope I didn't go too over. So thanks. Yes. Mm-hmm. Mm-hmm. And so maybe that's like, as we're getting older, we are more attached to models, but I feel like young people, younger people, like tend to update their models much quicker. And with that, maybe a similar measure of cognitive flexibility. Has anybody looked at that sort of comparison? Flexibility. It's a great question, and people have used the analogy of returning to a childlike state of mind to try to describe psychedelic experiences. And no one is, I don't know if anybody's directly made a comparison. I think it'd be a challenging comparison empirically to kind of tackle, but a really provocative one. And so that's, I think you're thinking in a flexible way that may really bear some fruit. Slightly reframing one of the things that you said is you know, relating to our models of the world. And so one of the kind of prominent, I don't want to call it a model because the way it's been presented is hard to test and falsify, but maybe an idea, theory, that psychedelics shift the weight of priors in a kind of Bayesian way. And that's something that really should be a lot more testable, right? And there are a couple of people thinking of how to use predictive coding paradigms to test that model. Yeah. And so I think you're on the right track. Yep.
I'm wondering if you can kind of expand on the idea of what happens during. What happens? Yep. Okay. What happens during the experiences that lead to the insights and the reframing? You sort of had a model there. And I'm wondering if beyond just the psychedelic kind of spontaneously creating these things, is there anything that you can cultivate in the therapeutic encounter to try to optimize that? Maybe especially in the context of non-responders?
Sure, yeah. So I think that most of the explanations of how the insights arise in the sessions is really a lot of weird kind of anthropomorphizing or beyond speculation, just storytelling. I'm happy to story. I love stories, but the analogies that are used by therapists who are not trained as empiricists come in the form of trust the wisdom of your inner healer, where your consciousness knows how to heal itself. You just have to get out of the way and let it, or things of this nature. Those I don't know how well those relate to or allow for speculation on biological mechanisms. The two or three most prominent models of the neurobiology of the acute effects or the acute experience. One is it kind of relies on the corticostriatal thalamocortical loop model that has been in some ways implicated in psychosis, I believe, at least at some point. And that model goes essentially, there are serotonin 2A receptors distributed and expressed along this kind of loop from thalamic nuclei to the prefrontal cortex back through the striatum into the thalamus. And that this loop model is one of the mechanisms broadly that may serve to kind of provide top-down or bottom-up control of stimulus information that makes its way to the cortex from the thalamus. And to the extent that psychedelics disrupt that circuit, they kind of disrupt the gate so that more sensory information flows to cortex, which further disrupts the circuit, which allows more to get through, which further disrupts the circuit in the feedback loop. And that leads to all the wild things that you see, hear, and feel when you are having these experiences. Now, that's not entirely satisfying for a lot of reasons. One, it doesn't really explain, well, what happens next? Or why would that lead to insight? Or why would that lead to an enduring change of being not depressed for a year or stopping smoking? And so I think you have to get creative in thinking then, well, what would the impact on modeling or on testing your models of the world or trying to kind of fit sensory information into models of the world? What would the impact of that be? And to the degree that you can convince yourself that would loosen priors, then maybe naturally insights will flow from that. Maybe it's an emergent property of disrupting the kind of landscape of energy in the brain or something like that, right? That's the best we have for now. Yep. Thanks.
Definitely bring more questions to the happy hour after.
So I promised some people that we would have free samples, but this is not California, so we can't do that. And but we get at Kellie's. Actually, my wife that goes to some of these conferences where it's actually true that I'm sure Fred's been to some of these as well. All right, so now we move to rats and rodents. So Kellie Tamashiro is a professor in the Department of Psychiatry and Behavioral Sciences. She's been here for quite a while. She's, I know Kellie from her work on other disorders, and so effective stress, especially early stress on eating disorders, obesity, and depression. And so, but I realized recently she's been started to look at psilocybin. So I'd like to know why and why you go from your sort of traditional path to psychedelics. So I'll turn it over to her.
Okay, is this working? Can you hear me all? Okay, great. Can you see me? Okay. Well, I just want to express my gratitude again and join Fred in thanking Tricia and Rick for having us here to share some of the work that we're doing in the psychiatry department. I think this is a great venue for sharing some of the research that's going on and to perhaps establish some new collaborations, and really to try to capitalize on the rich expertise and technological approaches that all of you are using in the neuroscience community here at Hopkins. So first of all, I just wanted to introduce myself, the lab. I'm part of the Division of Behavioral Neurosciences at the School of Medicine. And let me get my pointer up here. Okay. And so the group is headed by Dr. Tim Moran, and he has established this group here at Hopkins over the last, I think, 3 decades. I also want to introduce you to Zach Cordner, who is really the person who initiated these studies as a postdoc when he was in my lab. He joined the faculty and has since left Hopkins and moved on. But really all of the data that I'm showing to you today is from the studies that he initiated. And so that's your answer to why I got into this. It's primarily because of Dr. Cordner here. Okay, so like a good pharmacologist, a pseudo-pharmacologist in my case, we first did a dose response study in the mice that Zach had. And so what I'm showing you here is data from what's called the head twitch response. We tested various doses, injected IP into the mice, and Zach monitored the head twitch response, which is kind of like a wet dog shake. If you think about your dog coming in from the rain out here today, they kind of have this wet dog shake and see on their head. And I see some of the people out here exhibiting that behavior. And so the behavioral response, this behavioral response has been associated with activation of the 5-HT2A receptor, which Fred already talked about. It's thought to be one of the main targets of psychedelics. And really, it's kind of a screening tool to see whether or not we're actually engaging these receptors. And so what you see here on the right side is the head twitch response to 4 doses of psilocybin injected IP. And what we found is that there's a significant increase in these head twitch responses at 1 mg per kg. You see a decrease at the 5 mg per kg dose, and that might be primarily because of the sedation properties of this higher dose in the mice. Okay, so I'll note that this is really only a screening tool. We really don't know what this behavior or how it's associated with any of the behavioral outcomes. But it'll be important for some of the data that I'm going to talk about later on when we try to block the 5-HT2A and see what consequences on behavior. Okay, so next what we did was to ask whether or not we see changes in behavior in these mice. And unlike Fred's studies, we cannot do behavioral assessments in rodents by asking them about questionnaires or what their mood states are like or how a specific drug is acting on, is affecting them. And so we have rodent behavioral assays, and I know there are some students in the audience here, so I'm just going to take a moment to explain some of these assays that we're using. These are 3 behavioral tests that we are using in the studies that I'm going to be describing today. First is the elevated plus maze, which is supposed to measure anxiety-like behavior, and I say anxiety-like in quotes because we can't really know that these animals are anxious, quote unquote. But we can measure their response to this environment. Mice tend to stay in the closed arms, but their natural curiosity will make them go out into the open arms. And a normal mouse will go out into these open arms. A mouse that's been exposed to something like chronic stress will tend to stay more in the closed arms rather than venturing out. Antidepressant or anxiolytic drugs will actually increase the amount of time the animal spends outside. And the second test is what's called the forced swim test. This measures depression-like behavior. Basically, it's a beaker of water where we put the animal in. Most normal wild-type animals will try to swim around looking for an escape for a period of time until they start to float. And the time between the time that they swim and start to float The greater that time is, there's thought to be that less depression-like behavior. They're making efforts to go and find this escape, whereas the ones that will give up earlier are kind of just showing the kind of behavioral despair and helplessness behavior. And finally, we have the sucrose preference test, which measures anhedonia, or aversion to what would normally be a rewarding substance like sucrose. Mice really like sucrose, and so we give them 2 bottles of plain water and 1% sucrose, and a normal mouse will prefer the sucrose over the water by a ratio about 80% to 100%. With chronic stress or anhedonia-like behavior, we'll see a reduction in that sucrose preference. So what do we see in the behavior? So here are the animals that were injected again with the 5 doses that way I described earlier. This is elevated plus maze latency, and as you can see, we have a dose-dependent reduction in the latency for the animal to explore and go out into that big open arm. And we also see a dose-dependent increase in the full arm extensions. I'll note that this full arm extensions are a very rare occurrence, and in mice, I was honestly surprised to see that they were actually engaging in this behavior. And so they're showing some anxiolytic-like behavior, less anxiety with the higher doses of psilocybin. In the forced swim test, the latency to give up in that test increases with higher doses of psilocybin in here. And so what I'm showing you here is that at least at 2 to 4 days, there are significant effects in increasing anxiolytic and antidepressive-like behaviors. We have tested these animals out to 4 weeks and see that it actually persists out to at least 4 weeks in these animals after 1 single dose. And so for the next studies, we selected this 1 mg per kg dose that resulted in significant head twitches, but didn't appear to cause any type of sedation in the mice like the 5 mg per kg did. And so now that we have established that psilocybin has an impact on behavior, we next tested the role for the 5-HT2A receptors, and using this compound, ketanserin, which blocks the 5-HT2A and 2C receptors. And then we treated them prior to treatment with psilocybin. When we looked at head twitch behavior as expected, you see that there is little to no responses in the saline or the ketanserin group, but an expected increase in head twitch responses in the psilocybin group. But pre-treatment with ketanserin blocked this response. And so that made sense to us, and we asked whether we would see the same behavioral effects. And when we tested in the elevated plus maze, we were actually kind of surprised to see that actually the ketanserin did not attenuate that effect of psilocybin. And so this increased response may at least partially be independent of engagement of the 5-HT2A or 2C receptors. So these first studies were all done in naive mice. The next thing that we wanted to do was to see whether or not it was effective in a disease-relevant model. And as Rick mentioned, I study stress. We use several chronic stress paradigms in the lab. One is called the social defeat stress, which is a dominant-subordinate relationship between 2 mice. And the only limitation of this model is that it's generally only effective in male mice, because female mice do not show the aggression that male mice do when encountering another conspecific. So another model that we use in the lab is called chronic variable stress, or CVS. And it's a paradigm that usually lasts for 14 days and consists of relatively mild stressors listed here, and we administer them in a random order across days. And basically, it's a non-habituating type of stress. And so we can stress the animal every day. They won't habituate to this stress like they would for something like repeated restraint stress, where they know they're expecting the stress at a specific time. They know that, what we're going to let them out afterwards, and it's a safe environment. And so CVS is widely used in the field and reliably induces behavioral deficits that are relevant for studying mood and anxiety-like behaviors. And so we chose the CVS model. And what I'm showing you here is the head twitch response. Let me just orient you. First of all, we had 5 groups of mice here. We had a non-stressed, non-treated group in the first column here, and 4 groups of stressed mice that were treated with different combinations of ketanserin and psilocybin to determine what the role of the 5-HT2A receptor was again. And so we saw that, as expected, with psilocybin treatment, we see an increase in head twitches that was blocked again by the ketanserin pre-treatment. And so next, with the behavioral testing, we found that CVS stress, this is elevated plus MACE data. So the CVS stress reliably reduced the increased anxiety-like behavior, or reduced the amount of time the animal spent in the open arms. And at this dose of psilocybin, we recovered that behavior. And as you can see here, the ketanserin did not block the rescue of that behavior. And here is the sucrose preference test, testing for anhedonia again. And we see an expected reduction in the chronic stress group that is rescued again by psilocybin and not blocked by using the ketanserin antagonist. So we've demonstrated significant changes in behavior with the psilocybin treatment, both naive and chronically stressed mice. Zach's next question was, well, what's happening at the level of the brain? How is this actually having its effect? And so he worked with Jun Hua, who is at the Kennedy Krieger Institute. They have a preclinical imaging center there that has an 11T MRI scanner specifically for rodents. And we used naive mice, at least in this initial study. The mice were anesthetized, and they remained anesthetized for about 4 hours. We first did a 1-hour structural scan, then injected the animals, as I mentioned, described it before, with the 1 mg/kg dose of psilocybin. We waited 30 minutes, and then we started this eye vasal sequence, which is a measure that measures cerebral vascular blood flow and can be used as an indirect measure of neuronal metabolism and function. And so here are representative images from those vehicle or psilocybin treated mice. What you see here are warmer colors representing greater blood flow and cooler colors representing less blood flow. Quantification of those measures is shown here. And what we can see is that there was a reduction in the amygdala and hints of increases perhaps in the medial prefrontal cortex and the thalamic areas, with no differences in the hypothalamus. And so this suggested that there was some brain changes that are happening in the mice, even though they're unconscious in response to this psilocybin. But the question was, well, these animals are anesthetized. Does it still rescue their behavior? And so we tested them 3 days later in the elevated plus maze. And as you can see, we still had that rescue of the behavior, even though the animals were unconscious when they had that psilocybin injection and treatment. And so to summarize, naive mice can be used. We show feasibility in studying psilocybin, and we have behavioral changes that persist at least out to 4 weeks. In chronically stressed mice and a disease-relevant model, it's effective in reducing this chronic stress-induced behavior deficits, and psilocybin's impact on behavior might not be solely dependent on the 5-HT2A or 2C receptor activation, but there are some caveats to that we can discuss later. And finally, psilocybin treatment results in significant changes in the brain as measured with cerebral blood flow. And this occurs even under anesthesia, and the behavioral changes also occur in the animals, even though they were unconscious prior to and for 4 hours following their treatment. So these are very initial studies. We've established that we can use mice as to study the effects of psilocybin and perhaps mechanisms underlying the beneficial effects. we've established a collaboration with folks in biomedical engineering here to begin asking the question of how do psychedelics perhaps rewire the brain? How do they have these long-term effects on behavior? And Tim Harris and colleagues have developed what's called neuropixels, and some of you may be familiar with this, but basically it's in vivo high capacity electrophysiology. And so these probes are implanted into the brain. They can measure several thousand cells at one time. And taking all of that data together across time, we can measure neural activity, connectivity, synchrony at multiple points before, during, and even after the drug administration. It gives us the ability to record longitudinally over 2 months. And we plan to use different behavioral tests that we can use repeatedly across time to see how brains of these mice have changed and what might be responsible for the persistent behavioral improvements. And here, cognitive flexibility is one of those that we are very interested in, similar to what Fred had described. And so those are the data that we've started with and that we have right now. Some of the future directions that we're going in are going to be presented by Brianna Bullock, who's a postdoc student in the lab, very interested in what the environment is, what the role of the environment during and after the psilocybin treatment might have on beneficial effects. Does group housing versus social isolation produce differential behavioral effects? And MSA O'Donnell, who Fred also mentioned, is a collaborator that was working with Zach before he left, studying the effects of psilocybin on the microbiome and determining whether or not that might be one of the mechanisms that might impact brain function as well. And so to end, I just want to acknowledge all the people who actually do the work in the lab, many of whom are here in the audience. And I really want to acknowledge Zach Gordner, who's now with the Mind Work Group here in Baltimore, for spearheading all of these initial studies and kind of launching us in this direction. And of course, all of the different collaborators that we have, and look forward to any questions and discussions. Thank you.
Hello, cool talk. I was wondering, so you said you're doing 2-month continuous recordings? Doing 2 months worth of like constant recording? Okay, yeah, so like, cause I thought with NeuroPixels, you could, with several, you'd have to kind of keep them head fixed while you recorded. Do you have to do that in this case or no?
I don't think so. Austin Graves is probably more knowledgeable about the specifics that way, but we had planned to do behavioral experiments in awake behaving mice.
Yeah.
And so they should be patent. I know that we can get at least 2 in there. We're going to be ambitious and try to get 4 in there.
Oh, cool. And then I guess since the animals were anesthetized in with your MRI study,
Yeah.
I guess what do you think of the idea of, I guess, you having to have the experience for it to be important? So, because it sounds
Yeah.
like in this case, although then again, maybe mice don't have this or rats don't have the capability of having that experience or whatever. But
Yeah, I
like,
think
what do you
that's a
think
great
about that?
question. That was kind of my response. I don't know that mice have traumatic, early life experiences that come up and they can cope with, as some of the patients who have been treated with psychedelics kind of describe. We have a grand round session in psychiatry, and we typically bring in patients who describe their experiences with conditions and then treatments. And yeah, they describe, having these really profound recall of experiences or, visions of seeing their family members coming to rescue them, for example. And I'm not exactly sure that the mice, I don't know. You can. Good questions. Hi.
I was with the earlier talk too, and then yours, I was just thinking to myself, and even hearing about this work before, about what changes in the brain that in this fairly brief experience of a few hours has these long-lasting changes. And I'm just curious about what I'm sure you don't know the answer yet, but what are the current thoughts? Is it some massive synaptic plasticity that stays, or is it some change in gene regulation or expression, or what level are we talking about? It's like a switch that then just stays constant. That's a very different type of plasticity
Exactly.
than standard.
Can I say all of the above? We really don't know what's going on. And, these are initial studies in my lab. Other people have looked at synaptic plasticity and showing, spine growth in a very short time. We'd like to take advantage of some of the types of mouse models that Rick has to look at these exact measures, whether it's specific types of synaptic plasticity, specific genes, or is it just a whole brain reboot that, contributes to a kind of a resetting of connections or communications.
No best guesses at this point. It's just all open, or what they're willing to say.
That's a good question. Yeah, I mean, a lot of what we're doing right now is more hypothesis generating, just because we don't know exactly where to start. What I'm excited about with the neuropixels actually is that we'll do simultaneous recordings at multiple brain regions, so we can perhaps elucidate communications in sequence, or increases or decreases in activity between different brain regions, and in what kind of time scale that happens.
It'd be interesting to look at differences in excitatory versus inhibitory neurons, for example.
Mhm.
Yeah.
Exactly. Thank you.
Yeah, hi.
Okay.
Thanks. Great
Thank
talk.
you.
So the intriguing idea that there may be a dissociation between the psychedelic experience and the antidepressant effects is sort of kind of the elephant in the room. So I was just wondering if there have been any studies clinically in terms of like giving patients ketanserin to block the psychedelic effect and see if that.
Great question.
Yeah.
Actually, one of the first people to have done some of these studies is Scott Thompson's lab, when he was at the University of Maryland, showing that he could block some of the beneficial effects of psilocybin in a chronic stress model. And he has set up a clinical trial. Now he's out at University of Colorado, and they're proposing to pre-treat patients with ketanserin first and then administer the psilocybin. So it'll be really interesting to see what the outcomes of that is. I mentioned that there were some caveats to the ketanserin studies that we did. The dose we used was a 2 mg per kg dose. And I don't think it's blocking all of the 5-HT2A receptors. It may be enough to block the head twitch response, but it might be that it's engaging some of it in order to have the beneficial behavioral responses, first of all. And then there was another study that showed that there are 5-HT2A receptors inside of cells. There are intracellular receptors that I don't think the ketanserin can engage because it won't penetrate the cell. So I don't know what that has to do with all of this, but perhaps, those are the ones that are responsible for the behavioral effects while we're blocking others on the cell surface or something for the head twitch response.
This is somewhat related to the first question, but I was wondering whether people have looked at the beneficial effects of psychedelic, if it's given to the subjects while they're in sleep or under anesthesia.
Good question. I don't know. Do you know if they've tried doing that?
Yeah, so, there are a couple people at the University of Wisconsin-Madison who proposed both of those studies. I think they started with the sleep study and having people essentially provide IV or intramuscular psilocybin while people were sleeping to see if, I think they ran into a problem in the first couple subjects that kept waking up. And I think it's going to be an unscalable problem and experimental design. But the anesthesia question hasn't been directly asked yet with psilocybin, but there's an anesthesiologist and neurologist at Stanford or typhus who administered ketamine or saline to a handful of patients who were depressed and undergoing surgery for some reason. I think half of them had propofol, the other half were under one of the fluorines, whatever the case may be. It's actually interesting. They woke up after surgery and then on 3 separate occasions were asked to complete a clinician rated depression rating scale. And so after having told the clinician 3 separate times how they felt, then they were asked, well, you think you got ketamine or placebo. And of course, the people who felt better said, oh, I probably got ketamine, right? And people who didn't feel better said, oh, I probably got placebo, right? And there was zero predictive power in the estimation. And I have a lot of problems with this study. One of them is that let's acknowledge that anesthetics work by the profound effect in the brain, right? And so is there some interaction to expect drug-drug interaction? And then the way that we went assessing, what about assessing expectancy and predicting what they received? I guess I see some flaws in that study. People are proposing a similar study with psilocybin, but have not yet. There are whole companies that are trying to develop yes, exactly.
And that's rather controversial as far as I can. All right, great. So we now have food and drink, so please hang out, see the posters and talk to each other, and let's see if we can get 1 or 2 new collaborations out of the afternoon.